Good morning, everyone. Thanks for hanging in there. I know it's been a long conference, day 4 here. So I'm Matt Anderson-Barron, co-founder, CEO of Future Fields. It's really exciting to be here at PEGS. It's one of the few places you can come to and talk about protein expression challenges and the nuances of that, and everyone gets it because you've lived it, and it's really exciting to be here because of that. But today I want to talk about our platform, the EntoEngine, and specifically how we're using Transgenic Drosophila to turn what would normally be dead ends in your drug discovery programs into runways for development. So I'm sure everyone here in this room can probably answer this question. What project did you have to shelf because you couldn't access the protein that you needed? Maybe it was a candidate that wouldn't express, or quality was poor, or you just couldn't get the material you needed to do the studies that you needed to. Proteins are the foundation of nearly all drug discovery programs and biotech programs, and yet they are persistently stubborn and difficult to make. And because of that, they unfortunately kill a lot of programs, that are dying in the dark at the bench before the science really even gets a chance to scale. Now, when we step back and look at why that could be, and we look at different protein production workflow bottlenecks and actually where this breaks down, we can bucket these into a few key options. And so, first, there's a scale-up cliff. You have a protein that works at a small scale, but getting it to the quantities you need to do further studies can be a huge challenge, and it can be very expensive. It can take a long time. Two, you have expression challenges. Some proteins just don't want to be made. They can aggregate. You can get low yields, inclusion bodies, all kinds of challenges at the fundamental level of expression. Third, you could have issues with quality. Sure, the protein expresses, but the functionality is not there, or the purity is not there, or there's batch-to-batch variability. And fourth, when you look at the biological system, you can have issues with clonal stability, where you can get expression, you can get the biological system to scale, but over passages, the clones drift, productivity drops, and you're back to square one. Every one of these can derail a program, and they often show up in combination. These are not singular issues, but often overlapping. Because the reality is, every traditional platform has trade-offs against these bottlenecks. Yes, E. coli, they're fast, they're cheap, you can scale them like crazy, but for anything that's more complex, they struggle with a lot of the post-translational modifications. So typically you have to move to a mammalian system, something more complex. Transient CHO and HEX cells are great for fast turnaround of complex proteins, but volumes are very limited, and when you want to scale those, costs can start to add up very quickly when you need more material. So you can move to stable lines, stable mammalian lines like CHO or HEC to solve the volume and quality issue, but the price you pay is in time and cost. It takes a very long time to establish these clones, and stability with them can be an issue in themselves. The R&D investment is significant. So the message of this slide is quite simple. With traditional systems, you're always making a trade-off, and those trade-offs can dictate the success of programs or how those programs go, because you're having to choose between which box you want to check. And so that's really why we built the EntoEngine. The whole point of our platform is so that you don't have to choose, so that you don't have trade-offs, cost, volume, quality, time. The EntoEngine can check all boxes. And I know that's a very bold claim to make in a room full of skeptical protein scientists. So the point of this talk is to show that to you with data, with real proteins that we have made, and leveraging the advantages of our system. So first, what is the EntoEngine exactly? So it's a protein manufacturing platform that uses whole fruit flies. So specifically, we use genetically engineered Drosophila larvae as the production host. So instead of growing cells in a bioreactor, we have transgenic larvae that are being reared in these modular containers that express the protein of interest. So We've traded in the physics problems of the tank, like sparging rate and oxygen transfer and shear stress, for the resilience and elegance of a multicellular organism. And it's this organism itself that allows us to manufacture complex proteins better, faster, cheaper, and at scale. Now, there's a number of key headline advantages to our system that allow us to do what we do. First, we can be manufacturing ready in 28 weeks. So that means from plasmid design from the start of a project to having a scaled process that's ready for manufacturing in just over 6 months. It's predictable, reliable, and that matters enormously when you want to keep your programs on track. Second, there's inherent stability. So our strains themselves guarantee quality at scale, very consistent from batch to batch. Third, we have flexible batch sizes because of the modularity of our system that allows us to do short turnaround, and we can do everything from milligrams to multigram and eventually to kilogram quantities. And fourth, it allows us to unlock complex proteins at scale, particularly because of the multicellularity of our system. And so the rest of this talk is really structured around the 4 bottlenecks that I referenced and leveraging these advantages to solve those challenges. And I want to do that by showing specific case studies where we have made specific proteins for clients with problems that I specifically referenced. So starting with this scale-up cliff. So the problem is, you have successful screens that, die in scale-up. So you have a hit, you're doing a high-throughput study, you have a hit that works, the data looks great at the bench, and then you want to translate that into a process to generate gram quantities or more so you can do, in vivo studies, preclinical studies, whatever you need to do. And when you need to move to a stable mammalian line, that translation from the bench to that is kind of like a restart in the process. You have new clones, new optimization, new bioreactor conditions, new process development. The clock basically resets, and that can cause major delays. So our process with the EntoEngine, we move your protein from those high throughput hits at a small scale at the bench to manufacturing ready without restarting the clock, because it's the same process. It's a seamless transition. And I want to talk through this with a recent case study where we were able to do this for a client, where we took a protein from sequence to having it commercially ready at scale in 7 months. So looking at a quick timeline comparison, so the bottom track here is your kind of traditional CHO workflow, random insertion, selection, single cell isolation, et cetera. There's many steps in this process. Many of you are probably familiar with this. This taking 12 plus months is very realistic and can be quite optimistic. And we're saying this, we know this because a lot of the people that work for us now, they lived through that in previous roles, and that's part of the reason they were excited to come work with us. And the top track here is our process with the EntoEngine. We have very discrete steps. Everything's predictable, so vector selection, site-specific integration. You collect your stable fly strain, you scale it up to generate biomass, and you do your process development, isolate your protein, and validate it. It's all done under 7 months, and crucially, there's no re-optimization step in between the bench and the scale. So once we give you a sample, that process is ready for scale, and it's the same process we're going to replicate at commercial quantities. So it's not just faster, but it's predictable. So programs aren't going to stall here. And core to this is the modularity of our system. This is what our version of a bioreactor looks like. So once we've established a stable strain, scaling up is simply increasing the modules, increasing the number of ring trays that we're working with. And so that means a few things in practice. So there's no re-optimization required. The conditions that we're using to generate biomass for R&D or process development are the exact same for commercial production. So there's no translation loss. The protein that you're characterizing at a small scale is the same as what you get at large scale. So it's the same media, same infrastructure, same growth conditions. Everything is the same regardless of what scale, what protein you're producing. The bioprocess itself is copy and paste. The only thing you're really changing is the genetic cargo. And so scale up is exponential, and part of that is the modularity of the system, but coupled to the immense reproductive efficiency of insects, and it allows us to scale very, very quickly. So throughout this entire curve here, it's the same low cost, low maintenance system, so there's no inflection point or no big stepwise change where suddenly we need new equipment or new SOPs or new optimization runs. Once we have a process that's in these racks, in these modules, our ability to produce more, less biomass tailored to the needs of a client is very seamless, very low cost, very low maintenance. It's the same playbook, we're just replicating it every time. And again, critically, it's all protein agnostic. We do the same thing for every target that we are making. So, of course, scale up is meaningless if your quality drifts in the process. So this is a snapshot, this particular case study where we were making recombinant prolactin for this client that, again, we took from start to manufacturing scale in under 7 months. So you can see 3 different production batches here. The purity level is all above 90%. We produce in a glycosylated and non-glycosylated form of prolactin in this case. And you can see very consistent banding patterns across all 3 lots. And then when we test for functionality, in this case using SPR to look at the binding kinetics to the receptor, very reproducible curves, lot after lot. So not only are we doing this at scale, but the consistency from batch to batch is very good. So looking at the second bottleneck where you just run into a wall with expression. So sometimes it's not about time or scale at all. It's really just about getting the protein to express. And that could be for a number of different reasons. It could be toxic to the cells. It could be insoluble. It could aggregate. And that was actually the nature of our first real project with the client. So the client came to us to make BRA, the biotin ligase. When they tried to express it, they started with E. coli. Of course, it formed inclusion bodies that they couldn't deal with. They went to CHO cells to try that out, and the activity of the enzyme itself, when overexpressed, was actually stripping the media of biotin, so it was actually toxic. So, you know, you can have these 2 workhorses of recombinant protein expression, both of which failed to make this. So they came to us to make it in the EntoEngine, which we were really successful with. You can see on the right hand or on the left hand side there, the gel for BRA, very high purity, above 90%. And then when we tested functionality, we compared it to a commercial source, and we found that it actually outperformed in terms of function. So this was a protein, again, that these clients couldn't express at all, that we were able to get to work very well at scale with a high functioning, high purity version. So looking at quality challenges, you know, again, sometimes your protein expresses fine, you can get material out, but the quality is poor. You know, purity can be low, function can be compromised, consistency is poor, batch to batch variability, and to scale those problems often get worse, not better. And so we have a unique advantage here. Again, the fact that we're using a multicellular system means that the conditions with which we create these proteins, how we rear the insects, they can handle some perturbations to the system. You don't need to have these overly engineered conditions keeping everything tightly controlled, because if you change one little thing, all of a sudden your clone stops expressing or crashes out. You know, insects are incredibly resilient. They can handle variations. Of course, we do rear them under pretty tight conditions, but, you know, we also have a range of temperatures that we can use. You know, we have a range of humidity levels. There's not a requirement for these really tight engineered environments to get the process to work, to get a quality protein. Ours can handle, you know, some variability and still produce something that's going to look the same at the end of the day. And to talk about this, I wanted to use IL-12 because when we were here 2 years ago at PEGS in 2024, we had a lot of conversations about this protein. It was consistently referenced as a challenging protein to make for a couple of reasons. It's a heterodimer, so it's got the IL-12A and IL-12B subunit. Those need to come together, and they're linked by disulfide bonds. And importantly, they require the correct stoichiometric ratios to function properly. So here's some results from when we made it. So you can see in the gel there on the left, under non-reducing conditions, IL-12A and IL-12B, the heterodimer right around 60 kilodaltons in non-reducing conditions, and we were able to achieve good purity levels. But importantly, when we looked at functionality, we did, in this case, a bioactivity test, probing, doing a PHA-stimulated T lymphoblast proliferation assay. We see good activity, which is suggesting that the protein's fully folded, it's assembled, and it's functioning as it should. And there's a few different ways, a few different architectural choices you have with making a heterodimer like this. You can put both subunits on a single transcript with a P2A self-cleaving peptide. You can use separate promoters driving each subunit. But what's really cool about our system is we can do something where we actually put the subunits in separate strains on separate chromosomes, and then we breed them together. And so the offspring will inherit both subunits in a controlled, balanced way, and you get clean heterodimer assembly in the progeny. And so that's what we were able to do to have good success with this project. And that kind of genetic crossing, that tool, is more challenging or difficult to do in say a cell-based system, whereas this is a unique advantage of doing this with a whole organism. I mean, doing these crosses is just routine for us. This is something we do all the time. So when we talk about quality, so IL-12 is not just a one-off. We've used the EntoEngine to produce a wide variety of cytokines, all with consistent manufacturing grade quality. So here you can see IL-2, 10, and 12, all produced with good purity. You can see on the gels there on the left. And when we look at functionality, in this case, both with a LMR blue assay and SPR for binding kinetics, you can see good functionality, consistent with a functioning protein properly assembled. But quality is not just about function and binding kinetics. Of course, it's about safety and stability as well. So we routinely test for endotoxins and mycotoxins. This is something that comes up often as a concern with our platform because it's a whole organism. So we are testing for these things as well. And you can see in the purified samples for IL-2 and 10, both have low endotoxin levels, below 1 EU per microgram. Both were negative for mycoplasma. And we looked at stability as well. So the curve on the left here, we're looking at IL-2 produced in a fresh batch and a batch that was sitting in the freezer for a year. And we see very consistent overlapping curves suggesting that the function is conserved. It's a stable protein even after a year of storage. And then again, looking at lot-to-lot consistency, you can see IL-2 in the table there on the bottom. We get very similar sorts of specs for EC50 with SPR, endotoxins, and purity levels. So getting consistent batch to batch across IL-2 as well. So lastly, talking about clonal stability and the stability of the biological system itself, being what we believe is a really fundamental major bottleneck with traditional platforms. And I think one of the biggest hidden costs with protein production, with mammalian systems, building a stable clone is notoriously hard. It's slow, it's unpredictable, it can be a major cost driver, can extend timelines. And of course, transient systems can sidestep that, but you can't scale and build a long-term manufacturing program with transient systems, of course. I've already talked a lot about the predictability and reliability of building stable strains with our system, which we've already touched on, but what I want to talk about is the longevity of those stable strains and the long-term stability of themselves, something that we do see a really considerable advantage with. And so one of the reasons for that comes down to a unique genetic tool that we can use in Drosophila genetics, one that's been used for decades actually with researchers, and that's balancer chromosomes. So if you're not familiar with these, balancer chromosomes are these specially engineered chromosomes with these inverted repeats, and that suppresses recombination events with their partner. So if you have your gene of interest sitting on one chromosome and a balancer on the other, that gene of interest doesn't get shuffled out or silenced through genetic recombination or over generations. It's effectively locked in. And so in practice, that means that the risk of instability, it shifts from being this stochastic system to being deterministic, and we have control over it. So we're not just, crossing our fingers, hoping that a clone stays productive. We're using established genetic tools to essentially guarantee that that strain stays stable, stays expressing generation over generation, even at scale. And this is proof of that. Here's a Western blot from a strain producing prolactin FC. This was purified from a manufacturing strain that we have been continuously rearing in those racks for 3 years straight. So same strain, 3 years of generations, and you can see we're still getting robust expression of prolactin FC right where it should be. That's a lot of generations, and that's the kind of strain longevity that we can offer and gives you a lot of confidence to build these long-term manufacturing programs around. So we've talked about, these different bottlenecks, scaling up, expression at the bench, quality challenges, and clonal instability. And in each case talked about how using specific data with real programs that the EntoEngine can handle those, which is why, we can confidently say that the EntoEngine is able to check all these boxes across cost, across volume, quality, and time. So you don't have to give up, a corner. You don't have to make trade-offs with your protein production workflows. So here's why all this matters, just to wrap up. The point of what we're doing, the point of building a new platform isn't just to build a cool new platform. The point is that you shouldn't have to make trade-offs with protein production. The science, your science, the stuff you guys are doing is much too important for that. It has to scale. So don't let your program die at the bench. Don't let it die in discovery, because innovation is really only a breakthrough if it scales beyond the bench, if it can reach the patient or the end user, the customer, whoever it's designed for. It has to leave the bench. In our system, Transgenic Drosophila with our EntoEngine can help you get there. And that's what we're here to do. So if you want to learn more, please come find us at our booth today. We'll be there all day if you haven't already. We'd love to chat more, learn more about the things that you're working on and seeing if we can help. But in the meantime, thanks for your attention and time. Happy to answer any questions from the audience in the few minutes we have left. Thank you very much.
This is an amazing talk. I'm
Thank you.
Astonished. I used to be a Drosophila biologist, so I know some of the actually pains about doing that. So the one thing that when you talk about stability is there's something that we cannot do at the time. So I don't know if it improved. We could never freeze the embryos or the strains for us, because we as a grad student generating these transgenic lines, they're not easy to generate. After that, we have to have been mated and then to go on. And I lost quite a few important strains that way. So I wonder if that's a concern here.
Yeah, a great question. First, my understanding is there has been some work to show that they can cryopreserve the embryos and then still get viable organisms. We don't do that. We haven't had to yet, at least. But it's more of an operational challenge. So what we have seen, and we're for that kind of work, kind of generating our version of a master cell bank. Typically, we're just doing this at a really small scale, like maintaining, our one strain in a vial. Typically, as long as they're maintained inbred, typically the stability is quite good, and you're not going to lose it. And then you tip them, like give them kind of fresh food and stuff, about once a month, maybe twice a month. So it's really low cost. Of course, that adds up as you start to work with more strains. But for perspective, there are 3 different Drosophila stock centers around the world, one in Bloomington, Indiana. They house 70,000 different genetic strains, all simultaneously, that they're able to maintain in these little vials. And we know some of the folks from there, the director, And he's told us, we have strains that are like decades old, that are the same ones. We just maintain them, and they have a team of undergrads that just tips flies all day and keeps the machine running. So it's an operational challenge as it scales, but certainly doable. But great question.
Hi, thanks for the great talk. I have a question about the expression. So are you expressing the proteins in the whole fly or larvae all the time, or are you using UAS Skull4 lines to express specifically in tissues?
So short answer is both. Typically, we start with like the full every cell because you get better yields, of course. But there's the optionality to do tissue restricted expression. You can simply use different driver lines, cross them, and we have done that to varying degrees of success. It obviously affects your yield, but it is an advantage we have. So in certain cases where, let's say you needed a specific biological environment for a particular protein, we have a bunch of different cell types and tissue types to choose from, where you could just say express it in the brain or in the eyes or gut cells or whatever, to provide something that will function more correctly. But then of course you're gonna affect your yield. So yeah, so we've done it. We don't do it regularly, but it is a tool that we have in our tool belt.
Okay, thanks.
Thanks for the interesting talk. I had a few questions. One of them was about incubation temperatures. So is this something that you've tested tweaking, like when we did fruit fly research in one of the labs I worked in? We often had incubators at 18 degrees and then other ones where we just grew them at room temperature. Is that something you've tested in comparing like protein expression levels? Is that like a tunable variable that you might want to use, but also that might introduce an issue of equipment, like specialized equipment that you would need for these large racks?
Yeah.
I don't know if that's something that you've tested.
Yeah, it does affect it. Most of the time, like 25 degrees Celsius is optimal for the reproductive rate of the insects. We will use 18 if we want to slow it down. So it actually changes the life cycle from like 11 days to I think like 18 or something like that. So if we want to do long-term storage, small strains, we'll do it at 18. Normally it's 25. We can also use heat shock conditions for like inducible expression. All that's kind of built into our facility. So we have a manufacturing facility. And so we have different rooms that can be temperature controlled, environmental controlled for different programs.
Okay, and then one of the other questions I had was with the media formulation, are you using like a standardized media, or is that something that also has an impact on like the protein expression?
Huge impact, yeah. We've changed it dramatically. We took like kind of what was used in academia. What's used in academia is mainly meant for low maintenance. So they're actually designed to keep the strain's reproductive rate very low, because then it's lower maintenance. We didn't want that. We wanted high reproduction. So we've changed it quite a bit, not dramatically. It's still six simple ingredients. All of them you can get from a grocery store. So like supply chain is very robust. But yeah, it's quite a bit different.
Okay.
Yeah.
If somebody else has a question, I can stop. I can ask more later.
You can always come by the booth too. Yeah.
More questions. Too. Oh, I can't go that way. Oh, God.
Thank you for the talk. I have a question about like scalability, and I guess I don't know if you measure it in like trays or volume of the organism, but what's the scalability of this, and like I guess how many trays or rack systems do you need for if you're scaling up in the order of what would be thousands of liters for a traditional fermentation? Or
yeah, I mean that's a big advantage we have is the scalability. That was kind of what was in mind when we were thinking about this system. So we showed those racks. So all of it's modularized. So when we scale, it's just more of those. So picture like a server farm, like an Amazon warehouse where you just have rows and rows and rows of these trays. That's what we envision scale up looks like. And again, it doesn't require clean room conditions. It's more akin to like a commercial kitchen that you'd want for in terms of cleanliness. And that's even for GMP conditions. That's something we've confirmed with regulators. So the scalability is quite immense. The other factor we have too, these insects are putting off biomass every single day. So it's this cyclical process. So we're actually harvesting biomass, harvesting the larvae every day. And it can be stored frozen for very long periods of time. We've had biomass in the freezer for a year, and we've been able to take it, process it, get high quality protein out. So just supply chain wise and the robustness, resiliency of the system, it's quite immense because of that, because of the daily production, because of the ability to freeze as well.
We've got one last question and then we'll head to your booth.
Sure.
Thank you very much. Have you tried multi-specific to express, and how can I get the larvae?
How can you get the what, sorry?
Multi-specific antibodies. Can you express that?
We've not tried it. We could. We just need to find, you know, these days we produce what our clients want. We just haven't had someone come to us about that, but I don't see why we couldn't. Yeah.
And how can I get it in Canada?
Sorry, how can you get what?
In Canada.
I don't know. Yeah.
Okay.
Yeah.
Do we have,
No, I think we're, yeah. Was there one more?
was there one more? Well, yeah. And then, but I have one. Matt, what's your booth number? So,
Oh, 613? Yeah, 613. Yeah.
Okay, so maybe we'll have folks find you over there. And
Sure.
so let's thank Matt again.
Thank you so much, everyone.